Hi students, today's topic is estimation of urea. Before going to the estimation of urea, you should know about the normal level of urea in serum as well as urine. In serum urea or blood urea is 15 to 40 milligram per deciliter, whereas in urinary urea 20 to 40 milligram per day, that is 24 hours. So what are all the introductory points for urea? That is, what is urea? Main end product of protein catabolism. Where it is synthesized, we all know about the ammonia, which is very toxic in our body, which should be converted into non-toxic, that is urea in urea cycle. Automatically it will get excreted by kidneys. So the synthesized organ is liver, that is urea cycle occurs in liver. What is the structure of urea? One carbon which are attached to the oxygen through double bond and two amino groups, that is NH2 groups, right? And excreted by usually kidneys. What is the relationship between kidney, liver and urea? If the liver function test and renal function test is normal, means definitely the urea level also will be normal, right? Next one is, what is the significance of urea estimation? This estimation of urea is, it is a tool to measure the renal function test. It is a marker of renal function test. Okay. Next, what are all the methods available for the estimation of urea? We have classification, that is, one is enzymatic, another one is non-enzymatic. We are having only non-enzymatic method is diacetyl monoxide, right? Whereas non-enzymatic methods are Nestle's method, Bartlett method, urease glutamate dehydrogenase method, and a strip method, that is like glucose strip we are using. Urease coated urea estimation strip also, that is strip method, also sticks. Then the last one is fully automated analyzer, also we are measuring the urea, right? Next, coming to the DAM method, diacetyl monoxide method. Diacetyl monoxide. Here the first point is principle, second one is reagents, then procedure, next calculation, result and interpretation. These are all the subtitles for the DAM method. The first one is principle, how it produces color. DAM reagent combined with the urea, right, produces pink colored complex. That's all, very simple. In acidic medium, that is H2SO4 sulfuric medium. Here we are adding, that is ferric chloride and thiosemicarbazide. What is the role of this ferric chloride ions? Intensity of the color. Thiosemicarbazide, it acts as a catalyst. So with these three, the DAM reagent condensed with the urea molecule produces pink colored complex. After that, this colored complex will be measured by colorimetry, right? So the intensity of color is always directly proportional to the concentration of urea present in blood. Okay. So what are all the reagents for everything? Glucose, albumin, cholesterol, bilirubin, urea, creatinine. You have to mention the name of the reagents. That is blood sample, urea standard, DAM reagent and acid reagent. After that, what is the procedure? You have to prepare 3 test tubes. You take 3 test tubes marked, that is labeled as blank, standard and test. Blank means except analyte, it consists of all the reagents like distilled water, other reagents, no analyte. Standard means known concentration which will be prepared by your lab people personnel. Third one is test. This is patient sample. You are going to analyze the analyte present in the blood. That is, this is standard means known concentration, test means unknown concentration, right? So with these 3 test tubes, you have to mix well in water bath for 20 minutes. So you have to prepare 3 test tubes, blank, standard, test, add the mixture, that is reagents, wait for 10 minutes, then go for the water bath for 20 minutes. right? After 20 minutes, you have to cool the 3 test tubes, then go for the colorimetric estimation, that is reading, colorimetric reading. So after 20 minutes, after cooling, you have to take the OD at 520 to 540 nanometer, mostly 540 nanometer with the green filter. You have to measure the 3 test tubes, blank test standard, and you have to get the value 1, 2, 3, that is blank test standard values in your answer sheet, right? Actually, NPN is one important question asking some examiners in your university examination. Non-protein nitrogenous substances. Actually, the definition is these are a small water-soluble molecules, nitrogen-containing molecules that are not proteins. That is why this is known as non-protein nitrogenous compounds. Examples are urea, creatinine, nucleotides, and sometimes free amino acids. Right. They are synthesized both exogenous and endogenous. Endogenous means in our body by the protein metabolism, from the protein metabolism. Exogenous means through our diet. You are taking diet. With the diet, they are producing non-protein nitrogenous substances. This is very, very important question for your urea estimation. Second one is, you are getting the blank value, test value, and standard value. You are going to apply the formula and calculation, do the calculation. Finally, you will get the result, whatever result you write like this milligram per deciliter. After this result, you are going to interpret, that is clinical interpretation. We already know the normal value is 15 to 40 milligram per deciliter. So your value is suppose low or high. According to your value, you have to give the interpretation. Here we are giving some classification for high urea level. The first one is actually generally high protein diet always will give high urea level, right? Second one is one classification related to kidney, pre-renal, renal, and post-renal. Okay. In pre-renal, we have so many conditions like vomiting and diarrhea leads to dehydration, hemorrhage and hematemesis in the GIT tract or abdominal problems, or shock-like conditions, cardiac failure and burns. So these are all the pre-renal causes. What happens in pre-renal causes? How it leads to urea level high. That is decreased plasma volume. That is the main reason for this pre-renal causes. Decreased plasma volume that leads to decreased blood circulation. That is renal blood flow. That leads to decreased GFR. And finally, retention of urea. So this is the mechanism behind the pre-renal causes like vomiting, diarrhea, hemorrhage, hematemesis, shock and cardiac failure. And coming to the renal conditions like renal tuberculosis, malignant hypertension, polycystic kidney, renal failure and acute glomerulonephritis. These are all the causes for renal conditions. The next one is we are having post-renal, that is after kidneys. What happens? Stones in the urinary tract, prostate enlargement, prostatomegaly, then bladder tumors, tumors in the bladder, urinary bladder, and stricture urethra. So these are all the this is the classification for your high urea level, right? Next one is where you will get low level urea. First one is overhydration. Second is liver diseases like cirrhosis of liver. Third one is low protein diet. These are all the conditions which will produce low urea level. Whereas we are having two physiological conditions. Normally, pregnancy and starvation people will have low level of urea. Okay. Next one is urea clearance. After your result, you will go for interpretation. After interpretation, some examiners will ask like urea clearance, right? So you have to define what is the clearance, like creatine clearance, inulin clearance, and urea clearance. The definition is same. Clearance of a parameter like that through urine. We have formula and what is the result. Formula is U V bar P. You should know about what is U, what is V, what is P, and finally what is your result. Actually, for urea clearance, the normal range is 60 to 95, usually 75 ml per minute. You just know the result of the urea clearance, 75 ml per minute. Next, they will ask which conditions you will get high urea clearance, which condition you will get low urea clearance. High urea clearance means early stages of nephrotic syndrome, whereas low urea clearance in severe renal failure, that is less than 20% of GFR, right? After this urea clearance, they will be asking what is BUN, that is blood urea nitrogen. Sometimes serum urea is reflected in the nitrogen content in the blood. So 6 to 20 milligram per deciliter is the normal BUN value. That is BUN value is 6 to 12 milligram per deciliter. This is actually one of the indication. Actually, suppose the value is more than 100, more than 200 means one of the indication of renal problem, right? Here, sometimes the examiner will ask what is the conversion factor for BUN to urea. That is 2.14. That is BUN value into 2.14 is equal to urea. Urea is equal to BUN value divided by 2. Like that, the conversion factor is 2.14 is for BUN. Okay. Next, you should know about the difference between urea clearance and creatinine clearance. Which one is best, which one is not usually routine. Like that, some examiners will ask. Always creatinine clearance is the best one. They are having some futures of creatinine clearance. You should read about both urea clearance and creatinine clearance. The last one is estimation of urinary urea. The same method, DAM method. Normal level is 20 to 40 milligram per day. Decreased in acute renal insufficiency and increased in pre-renal conditions like hematemesis and hemorrhage. So this is regarding urea estimation, right? Two questions for your homework. One is what is protein-free filtrate or how you prepare protein-free filtrate. Second one is what is azotemia and what is uremia. What is azotemia? What is uremia? Right?